In this video, I'm going to present our recent study using mutational signatures to uncover the role of smoking in COVID-19 vulnerabilities. I am Yoo-Ah, a staff scientist at NCBI. Human genomes accumulate mutations over time. But most of the mutations are unharmful passenger mutations that do not cause any diseases. Nevertheless, analyzing the passenger mutations can provide us a valuable information as different metagenic processes generate characteristic mutation patterns. Some metagenic processes are environmental, such as smoking, or the exposure to UV sunlight. Some are endogenous, such as DNA repair mechanism dysfunction. In this study, we utilize mutational signatures to understand the impact of smoking and other environmental factors on COVID-19 infection and severity. To this end, we took the lung cancer dataset from TCGA and extracted mutational signatures using the mutation data from cancer tissue. Even though we extracted signatures from cancer tissue, we expect environmental factors such as smoking affect the whole individual regardless of tissue types. For gene expression data, we use the RNA-seq data from normal samples to remove the effect of cancer. We then computed the correlation between the activities of mutational signatures and the expression levels of SARS-CoV-2 entry genes. Even though some of the correlations are not statistically significant, possibly due to the moderate number of samples, we found that the expression levels of many genes are positively correlated with smoking signatures, as well as other mutational signatures, suggesting the increased risk of virus infection. We then expanded our analysis using ECCB algorithm. ECCB algorithm is a simple but powerful clustering-based method to uncover biological processes associated with mutational signatures. Using ECCB algorithm, we identified two clusters which are positively correlated with smoking signature. One cluster is correlated specifically with smoking signature and is enriched with the genes related to cytokine-mediated signaling and inflammatory response. The other cluster is positively correlated with all the signatures, including smoking, and is enriched with genes related to innate immune response. For both clusters, we found that the clusters are significantly overlapping with the upregulated genes in severe COVID-19 cases. And same for the innate immune cluster. Finally, To understand where the expression changes come from, we deconvoluted the RNA-seq bulk expression data using CyberSort-X. For epithelial cells, we found that the number of goblet cells is positively correlated with smoking signature, suggesting the inflammatory response from epithelial goblet cells. For immune cells, innate immune cells such as neutrophils, macrophage proportion is positively correlated with all the signatures. In conclusion, we found that the gene expression level of many SARS-CoV-2 entry genes have moderately positive correlation with smoking mutational signatures. We also identified two clusters of genes with strong positive correlation with smoking signatures. The clusters are enriched with inflammatory response and innate immunity, respectively. We also found that the clusters are significantly overlapping with upregulated genes in severe COVID-19 cases, which suggests negative impact of smoking on COVID-19 progression.